Yeah, I apologize for the small technicality, the technical issue that happened. So this is basically the last section of our course. We cannot finish our course by addressing of what exactly happens in the world in the context of the variable artificial kidney development. So it will be more like an interactive lecture. So we will try to remember what particular topics did we cover during the development of this course, during the duration of this course. So we started this course by actually discussing the main functions of human kidney. We discussed the structural unit of this particular organ that performs the blood filtration and reabsorption of the essential nutrients and water. Perhaps you can even name this particular structural unit. Can you name what is the structural unit of kidney? Nephron, yeah, correct. And basically it's the unit that performs the initial blood filtration, the water reabsorption, etc., and so on, including secretion and excretion, the waste urine from the human organism. And does the kidney have any other functions beyond the blood filtration and water level control?
Homeostasis?
Yeah, homeostasis is the part of the blood filtration process, but how is the blood filtration is the only tool for the homeostasis that?
Hormone regulation.
Yeah, hormone regulation, correct. That's endocrine function, and hormone regulation is a very broad topic on itself. We did not cover it in the details because it's basically the kidney function is the like a Ding and Zich. It's a thing that requires like the entire course that to cover all specific processes and physiological phenomena that take place in this organ, because kidney can regulate the calcium concentration and calcium metabolism, and can regulate the hemostasis function, etc., and so on. And of course, when we talk about the chronic kidney disease, it's not just about the blood filtration. The blood filtration is the primal concern, because without the blood filtration, the patient will develop the uremia, and the level of toxins that increases in the human organism will lead to very severe symptoms, and at the end it will lead to the death of the patient. That's why the patient will require the renal replacement therapy in order to purify blood from urinic toxins, to excrete this excessive water and the urinic toxins from the human organism. And can you tell me what is the golden standard of the renal replacement therapy in your opinion?
Kidney transplant?
Yeah, it's an organ transplantation, that is correct, because at the current stage of the technology, unfortunately, there are no any medical systems or any apparatuses that could replicate or imitate all functions of the kidney at this point. It's usually revolves around the idea that we can filtrate blood from the excessive water and the urinic toxins that accumulate in the human body during the development of the chronic kidney disease. And people who actually are under the hemodialysis procedure or peritoneal dialysis procedure, at some point they even require the hormonal therapy, and they require some, in terms of hemodialysis, they require the additional replacement solution in case of the intensive ultrafiltration procedure. So during this course, we were talking about the different perspectives and the different developments of the portable or the improvement of the renal replacement therapies that we currently have. So we were talking about the just a small refreshment of what exactly we discussed. So it's not just like a development of very groundbreaking technologies like wearable artificial kidney or implantable artificial kidney. First and foremost, like the most desirable at this point type of technology would be the portable artificial kidney for the exact reason that we already possess the necessary technologies to actually implement the home dialysis at the patient's home without actually a requirement to go to the specialized dialysis centers. So if you actually check the statistics, Europe, specifically Germany and Scandinavian countries, they have a priority to actually make it possible for every patient who suffers from chronic kidney disease to perform the home dialysis procedure. For that particular reason, a development of the portable devices is required, and usually they divide these different portable devices into two generations. So the first generation was more or less bulky, not as big as the conventional hemodialysis apparatus that you can see in the hemodialysis center. But usually it weighed about from 10 to 15 kilograms. And the second portable devices generation was a miniaturized version of the first generation, so they weighed about from 1.5 to 3 or 2 kilograms, so it's a smaller version of the portable dialysis machines. There is still an issue speaking of these portable devices, probably speaking of the hemodialysis, you can even mention or emphasize that problem. Yeah, the primal concern, if we talk about the hemodialysis, is actually a water supply. And in order to perform a hemodialysis procedure, if we talk about the conventional dialysis methods, it usually goes for 120 to 150 liters per one patient. And at the same time, the production of this volume of the dialysis fluid requires even a bigger amount of water that has to be specifically prepared for the dialysis for the medical purposes. It means that it requires very specific parameters in terms of the bacteria concentration, in terms of the specific conductivity, in terms of the pH, etc., and so on. So no matter if we're talking about the conventional dialysis, hemodialysis, or we are talking about the portable devices that perform different modalities of hemodialysis at home, it still requires a relatively decent amount of dialysis fluid that we have to spend on one patient. And at the same time, if we want to prepare the sufficient volume of the dialysis fluid that can be used for one patient, it will require even a bigger volume of the water that we have to prepare. So in order to perform this particular procedure safely for the patient, we have to take into consideration that we need to establish the water preparation system or water preparation unit specifically to perform this procedure at home. Or at the very least, we need to supply patient with the prepared amount of the dialysis fluid to perform this procedure. For instance, if we just shift from the hemodialysis to peritoneal dialysis, we still have like option in Russia, for instance, in specific dialysis centers, they can perform the automated peritoneal dialysis procedure right in the hospital, or this automated peritoneal dialysis system can be established at the home of the patient. Depends on if the patient can afford this. So it can be a huge issue for some patients who require this procedure. So in terms of the home peritoneal dialysis, patients are usually supplied with the peritoneal solution, and per one procedure they have to spend about from 10 to 15 or even more dialysis fluid that is required for that. So basically it's a very consuming in terms of the water and the different supply and different details that have to be replaced after each procedure, such as in terms of hemodialysis, we need the extracorporeal mass exchange unit called dialyzer that is basically used for the blood filtration, and it requires it's a one-time used, usually it's a one-time used unit that should be replaced after each procedure. And this technology itself is quite expensive. That means that the manufacturing of this particular dialyzers is quite time-consuming, and it's an expensive process. And you can find some patents about the attempt to regenerate these membranes for the further usage, but the easiest and safest option for the patient to operate the hemodialysis at home is just to change the mass exchange unit. So basically it creates some specific issues with the conventional hemodialysis method, and while portable artificial kidneys, they basically have an opportunity to increase the social mobility of the patient, because he is not required to go to the dialysis center to perform this procedure every 3 to 4 days. He can perform hemodialysis at home. Nevertheless, he's still restricted by the home dialysis apparatuses. And another issue, if we talk about renal replacement therapies outside the kidney transplantation, what is the core issue we can emphasize in that particular case? If we compare the natural kidney to the automated peritoneal dialysis apparatus or the hemodialysis apparatus, what core difference we can actually notice here?
National kidney operates 24 hours.
Yeah, that's exactly a correct answer. Kidney provides a continuous blood filtration in comparison with the conventional renal replacement therapy methods. So it's not just about the social mobility, it's about how exactly we treat the patient. If the treatment that we provide is actually adequate enough for the patient, because unlike kidneys, in terms of the hemodialysis, we can treat blood only for a very short period of time. And another problem is that in terms of the hemodialysis, the only part of the cumulative water volume that we have in the patient, we can treat only blood. So we can access only the blood system, and we can treat it within the mass exchange unit. And that's a problem on itself, because if we have very intensive hemodialysis procedure, and if we decrease the concentration of specific, usually small-sized urinic toxins like urea or creatine and uric acid, what particular phenomenon can we observe in the blood after the treatment? Yeah, let's imagine that we have the concentration dynamics. So we have the concentration of some substrate before the procedure, and it rapidly decreased during the procedure, and that's some time when we end the hemodialysis. What do we observe after that?
Rebound?
Yeah, the rebound, so we will observe some sort of a slight decrease of the of the toxin afterwards. And this process can happen for 1 hour or 40 minutes. It depends on the patient's physiology. Can you provide an explanation of why it happens?
Because of the intracellular and extracellular solutions which are there in the intracellular and extracellular space, they move to the blood.
Yeah. So since we can provide like a simplified idea of how the water compartments presented here, we still have different compartments that we cannot affect directly during the hemodialysis. That means that if we directly interacted with blood and we decreased rapidly the concentration of the toxins of, for instance, urea, it does not mean that we actually interacted with other water compartments. In fact, we do not any have direct connection to this compartment. And what happens is when we decrease the concentration of urea, we remember the Fick's law, we create the gradient concentration between the blood and, for instance, intracellular fluid, and we observe the diffusion of the metabolites from one water compartment to another, specifically blood. And that's what we observe in the form of the rebound. So it's like a complication in terms of the hemodialysis if we talk about the physiology of this process, while kidney provides a continuous blood filtration, and that means that we have the even filtration of other water compartments in the human body. Hemodialysis is usually restricted in that particular case because we have a very severe time restriction. The patient can perform hemodialysis only for a very short time period, usually it's 3 to 4 hours, and he can perform this particular hemodialysis procedure only in the specific times of the week, usually 3 or 4 times a week. And while it's uncomfortable, so it restricts the patient mobility and social mobility, and it, at the same time it does not provide like a continuous blood filtration. That's why the further development of the renal replacement apparatus started to emerge. And usually when we talk about different approaches that we discussed during the course, if we just look at the historical background, for instance, enzymatic method that was implemented in regenerative dialysis called READY, was firstly implemented, if I recall correctly, somewhere in the 1980s. So it's basically a very old idea that has been tried to implement it in the form of the variable artificial kidney devices. But the interesting development of the technologies is that we have the idea, we have the first implementation of that idea, but usually at the early stages of these technologies, people do not possess other types of technologies that would provide them to implement the technology in a variable stage apparatuses or the implantable artificial kidneys. So it's very important to develop different branches of technologies that will provide us the opportunity to develop such next stages of the renal replacement therapies, like the variable artificial kidney that requires the miniaturization of the hemodialysis apparatuses, or the emergence of different sensors that provide the quality control, etc. And in case of variable artificial kidneys, we require the development of specific membranes that will provide us the opportunity to create the micro hemoperfusion systems, usually made in the form of a MEMS technology, or in the form of the more in the form of the U-shaped filtration units, like it was realized in a kidney project. So basically what has to be emphasized is that some ideas, they exist for a very long time. For instance, if we talk about the bioartificial kidney, you will probably recall the type of technology that was implemented for hemodialysis in attempt to imitate the natural nephron. Do you remember that? Sorry for that. Someone is looking for me, but I'm busy. Yeah, we were talking about the renal assistant device. It's basically it's relatively old idea on itself. Like it started to be implemented somewhere in early 2000s, and we were talking about this particular system. So, yeah, this particular method, as far as I remember, was described in 2008. While the idea itself is even older than 2008, but the first implementation was made actually somewhere in this time period, and basically that was an attempt to imitate how the nephron operates. So we have the hemofilter that operates basically as the Bowman space capsule that receives the primal ultrafiltrate that we send to the modified the hemodialysis that actually has the endothelium kidney cells that provide the reabsorption of very essential nutrients for the patient. So this system returns the blood to the patient with the nutrients that were transported from the ultrafiltrated back to blood, and then the spent ultrafiltrate goes to the waste. So basically that was the main idea behind this artificial nephron technology. And many prototypes of wearable implantable artificial kidney, they utilize the idea of the combination of some hemofilter and the combination of bioreactor that imitates the proximal convoluted tubule in that regard. And considering that this technology existed in 2008, you can imagine why exactly it was not implemented earlier. Why it actually took time for other implantable kidneys to emerge. Any ideas? I'm sorry. Yeah, the primal idea is the miniaturization of mass exchange units. So if we have the conventional hemofilter, it's actually a very huge column with membranes, and it could be even. It actually possesses a very big size, and unfortunately I had no opportunity to bring it with me for the illustration. But if we're talking about the conventional mass exchange units, they have the issue with the size of those units, and another issue is still a calculation problem. So in order to operate the hemofilter and the hemodialyzer, you require a heparin infusion, which is a problem in itself. So the conventional mass exchange units have a number of problems that have to be solved before we acquire additional level of technology that will provide us to create a miniaturized version of this particular renal assistant device system. That means that we need to create the MEMS technology that would be able to imitate the hemofilter device, and at the same time, since we are dealing with the blood clotting issue, we need to develop the sort of the protective surface that will be bioinert to the in terms with the blood connection, because as we discussed within the second tutorial, the biggest concern with any implantable device, no matter if we're talking about the artificial heart, or we're talking about bioartificial kidney, or we're talking about total artificial kidney that does not exploit any culture of human cells to imitate some specific processes within the apparatus. The primal concern that we have a direct interaction with blood, and usually the organism is smarter than we. If we assume that some specific material that we developed is completely biocompatible and it's completely safe for a human body, and it's completely applicable in terms of the implantable artificial device. What actually happens is that blood has the ability to recognize that the type of material that we introduced in the system is not natural. It's not like the original tissue that was before. And even if there is a biocompatibility of that material, there is always a chance that the blood clotting formation will start. So in that terms, the achievement of the biocompatibility, of hemocompatibility, is a very huge issue and a very huge concern that had to be resolved only relatively recently, because the bioinert materials started to emerge. And when they started to emerge, then the creation of the implantable heart devices actually became an opportunity that people could realize. So, speaking of the main kidney projects, like if we're talking the implantable devices, well, in terms of this lecture, we can actually divide them on the two different approaches. So we have the implantable artificial kidney, and we have the implantable bioartificial kidney, and we have the prototypes that we can, for simplicity reason, describe as the total artificial kidney. Implantable artificial kidney. So, as I mentioned before, the type of mistake I made in the lecture 5, if I recall correctly, when we were talking about the concept behind the implantable artificial kidney, I made a rather broad generalization that when we talk about the implantable artificial kidney technology, we usually talk about the dialysis-free technology. So it's basically an attempt to avoid the usage of any artificial dialysis buffer solution to purify blood. So basically the core concern here was an attempt to imitate the nephron as the structural unit of the kidney. Well, the mistake, why is that exactly a mistake? Because if we talk about the implantable artificial kidney technologies, and specifically when we talk about the prototypes that are suggested and proposed on the market of the kind of implantable artificial kidney systems, there is an attempt to create an implantable artificial kidney device that actually utilizes the hemodialysis. That's the part that we actually covered in the lecture 11. But here we will have the small promotion video that will basically illustrate what kind of achievement what kind of functionality they are trying to achieve within their particular device. Right now we're talking about one of the most prominent projects within this particular topic. It's the kidney project that is developed in San Francisco right now, and it utilizes the biotechnology as the main tool to achieve the major function of the reabsorption of the nutrients and reabsorption of the water. So the primal idea behind this particular tool is to create a device that will imitate the Bowman capsule space and the proximal convoluted tubule. So they have the sort of filtration unit that consists the biotissue deposited on the specific membrane, and they have an additional membrane to actually separate the blood from this particular biological material. Perhaps you have an idea why exactly they decided to make that move. Why do they need to separate the biological tissue from the human blood with additional membranes, so there will be no direct interaction between the biological tissue and the... Yeah? Prevent what? No, to prevent blood clottings, you could just basically use a bioinert surface to deposit it on the silicon nanomembrane, for instance, because the blood clotting would occur even if there were no any biological tissue and we had no means to prevent that. So the prevention of the by blood clotting and the achievement of hemocompatibility is a completely different issue in comparison to what we're discussing here.
Stop immune response?
Yeah, it's in order to stop the immune response. Another issue that we have here, even if we just take this cell culture from the patient himself and we use this sample to grow the biological film or the biological tissue on the specific substrate, even if those particular cells were taken for the patient himself, there is still a big chance of the immune response. So to decrease the chance of this response, they decided to apply additional membranes that basically separate the blood flow from the biological cells. So it basically helps them to prevent the main issue that occurs during the usage of the biotechnologies is the immune response of the human organism. And basically, I'm not sure if we have this illustration here. Yeah, probably we do not have. Yeah, we can just talk about this. So we have the hemofilter, we have the bioreactor, and this ultrafiltration usually goes to the bladder, and that's basically the idea that we implant this kidney in a patient's organism, and it operates like a natural kidney. Well, maybe with some restriction, but it's better than nothing. So yeah, the primal concern is the application of the biotechnology, which basically makes that a more time-consuming process to make this technology, and there are a lot of complications that might occur with that problem. Nevertheless, it's one of the main projects that are basically presented within the future market of the implantable kidneys, and probably, if I will be allowed, we can watch the short video provided by the developers of this kidney. Yeah, can you play this video, please?
After a decade of engineering development to refine the hemofilter and bioreactor components, preclinical prototypes of the implantable bioartificial kidney have been successfully created. Clean room microfabrication processes have been established to reliably produce high-quality silicon membranes. Tissue culture protocols have also been optimized to reproducibly grow renal tubule cells and maintain their function. The silicon membranes were mounted in polycarbonate housing to construct hemofilters, and in similar fashion, bioreactors were assembled to immunoprotect the renal tubule cells. The hemofilter and bioreactor components were then connected to create bioartificial kidney prototypes for benchtop and preclinical testing. In proof-of-concept experiments, the hemofilter operated under blood pressure alone, without systemic anticoagulation, while the renal cells in the bioreactor remained alive and healthy without immunosuppression. Further improvements will scale up the prototypes with additional silicon membranes and renal cells, creating clinical devices that can demonstrate sustained treatment of kidney failure in human trials. The implantable bioartificial kidney will provide the therapy and mobility patients need to move beyond the burdens of dialysis and organ shortage, and once again enjoy the freedoms of everyday life.
Yeah, thank you. So yeah, that's basically what.
आपके हिसाब से आजकल लोग सबसे ज़्यादा
Yeah, The YouTube advertisement is our everything. Yeah, basically what's prominent about that object is they already have the preclinical trials that they use the animal trials to actually test how this particular kidney will work. Well, in my humble opinion, if we're talking about the implantable kidney technologies, right now the kidney project stays closer to the further development and to the probable introduction of the kidney project into the market of the renal replacement therapy devices. They showed some essential things that have to be emphasized again within this course. They had to develop specifically for the realization of this project a new type of the silicon nanomembranes that would perform the blood filtration, or will be used for the blood filtration. That means that we make an emphasis for the creation of this particular implantable artificial kidney technology. We need a brand new membrane that will prevent the blood clotting, that will provide the blood filtration within the functioning of the human heart. So yeah, one of the core features of this particular project is that they do not require any additional pump installed in the inside of the human chest cavity or anywhere else. So basically, they utilize the natural cardiovascular system of a human patient to maintain the blood filtration just like it happens in the natural organ. And just to emphasize the interesting part of that project is that they already have the animal trials, and they utilize that particular device within the cow animal module for at least 7 days, if I recall correctly, with very positive results. So there were no immune response to this particular apparatus. It performed quite well in terms of the blood clearance and the urinary toxin clearance. And the primal concern that they are trying to address right now is the further manufacture.
So in the video, I may have heard wrong.
Yeah.
Did they just mention that they don't use any anticoagulation measures?
Well, usually they do not cover this part within the short promotion video. I think that they took a concern of what particular materials they have to use and what particular construction of this device they need to take into consideration to change for instance, the shear stress of the blood flow, the way there will be no clot formation. Usually promotion video does not cover all technicalities of the apparatus construction, and especially when there is multiple projects they try to develop their own implantable artificial kidney, they might not cover it just for a purpose.
I remember they said something like without any anticoagulation measures that their membrane was like intact after the trial or something similar.
Well, they still take the clot measures into consideration. It could be related to the manufacturing of silicon nanoporous membranes that they use. Perhaps this particular membrane technology already works as the bioinert type of a membrane with the lower probability of the blood clotting. But in those devices, just like we covered that topic in the tutorial too, the blood clotting issue is the critical problem that has to be addressed. And one way or another, they address that topic within the development of the kidney project. So some technicalities, they might be hidden in the patents or the articles, but that's just a promotion video to give a better outlook what particular prototypes exist within the market. Yeah. So yeah, I just started with this particular apparatus because it's one of the big fish of the prototypes of the implantable artificial kidney, and if we want to observe what exactly happens in the development cycles right now, I think the good start is just to read the articles about this project or to observe what current state they have at this particular moment. Yeah, returning to the bioartificial kidneys is not the only prototype that exists. There are attempts to create the bioartificial renal epithelial cell system that basically has to imitate the work of the proximal convoluted tubule. So, considering the historical perspective of this problem when we talk about implantable artificial kidney, and considering the success of the system that was the renal assistant device, it's logical to suggest that the first idea that goes into developers' heads about what particular implementation they can have in this particular issue is an attempt to recreate the renal assistant device in terms of the implantable artificial kidney system. And this basically was the next stage of this idea. So it's a bioartificial kidney where they further modified the bioreactor to the point that they created a system that could synthesize the bone morphogenetic proteins that basically they suggested it to use in a regenerative medicine as well. And speaking of European Union, so this is the American project, the project that has been developed in San Francisco, if I recall correctly. It's like not the only project in terms of bioartificial kidneys that is currently been developed around the world. Another attempt to create the bioartificial kidney, again, is an attempt to imitate the rat system within the implantable system, is the KidNew project. It's a European consortium of different countries of the European Union that are trying to create the implantable artificial device that would work similarly as basically the kidney project. And just to emphasize the scale of these projects, it might be very confusing that such a small apparatus like Kidney on Chip project requires the effort of many consortiums and many countries. The same thing you can observe in wearable artificial kidney technology. For instance, the AVAC system, automated wearable artificial kidney, is basically a consortium of Singapore and some European countries that participated in development of this wearable artificial kidney technology. And the same idea of these projects actually happens in terms of implantable artificial kidney. So it's not just the effort of one European country, it's the effort of many consortiums from France, Germany, and other countries that contribute in the development of this particular project. And basically it tries to utilize the same idea. So we have the hemofilter, the nanochip that provides the information of the ultrafiltrate that goes to the bioreactor with the cells that imitates the work of the proximal convoluted tubule. And that's the idea they are going to implement during the animal trials. And the main concern is, of course, the miniaturization and the achievement of the effective blood filtration within the miniaturized system. So, as we mentioned previously, the main concern is the development of brand new membranes that will allow within the system to effectively filtrate blood. Because when we talk about the implantable artificial kidneys, usually introduction of additional pump would be a very complicated issue that would revolve around the idea how to power up this pump or how to implant this pump safely for a human, for a patient. So in that case, the most obvious idea would be use the heart as a natural pump that would recirculate this blood through the implantable artificial kidney device. So for that matter, you need to develop the membranes that would provide the efficient blood filtration flow in order to achieve parameters that would be close enough to glomerular filtration rate of the natural kidney. And of course, the imitation of proximal convoluted tube is a must in that technology, because if you just filtrate blood, that means that you remove excessive water from the human organism, and that means that potential dehydration depends on the properties of the apparatus that you make. So the primal concern is the reabsorption of the useful components for the human organism, and the very hard technical issue is, of course, the development of water reabsorption. And usually, considering the historical perspective, the first of the solution was to try to imitate the renal assistant device system. So it's basically the same idea implemented in different projects across the different parts of the world. Well, speaking of the problem that we concern the total implantable artificial kidney devices within that course as well, that's the type of the prototype, the implantable attachable artificial kidney that is basically developed in South Korea right now. And that's a very interesting, in my perspective, project, because originally they were making membranes for the water filtration for industrial purposes. And they actually made a new branch of the technologies that they are trying to implement in the medicine, and they already implemented different membranes for the hemodialysis, as far as I know. And they grand object, their potential magnum opus, so to speak, is an attempt to create the implantable kidney device that would not require the usage of biotechnology, but at the same time it operates in a very interesting way, because I will repeat myself again. When we start the lecture 5, I was talking about the attempt to imitate the basic functions of nephron, and my broad generalization made like a delusion that all implantable technologies just go for the nephron. While this technology, if we watch the promotion video, they actually try to implement the hemodialysis approach within the implantable system. So if it's possible, please.
Travel, eat and drink freely, and return to a healthy life.
Can you just start from the beginning, please? Here. Thank you.
End-stage renal disease, so-called ESRD patients receiving hemodialysis treatment visit the hospital more than 156 times a year and undergo dialysis treatment for more than 4 hours to maintain life. Because of this, they are experiencing great difficulties and restraint in their daily lives, including work and hobbies, even eating and drinking. The biggest reason why patients suffer is because the current hemodialysis treatment was established in the 1960s and has been systematically maintained without any significant changes. Due to this system, approximately 1 20-liter of sterilized purified water is required for one hemodialysis treatment. Therefore, treatment is only possible in special environments, such as dialysis centers. Patients' lives are greatly restricted compared to healthy people, a situation that can happen to you at any time. Could you really live like this for the rest of your life? Unisex Pure Air proposes a new concept of hemodialysis system. Pure AAK stands for implant, attachable artificial kidney device. Pure AAK is a small artificial organ that replaces the kidney function of patients with ESRD. It receives blood containing waste products from arteries, purifies it, and then discharges it into the veins. The filtered waste is concentrated and discharged into the renal tubules to perform the kidney's main functions of blood filtration and water regulation. The composition is divided into 3 parts. This part mainly consists of 2 parts. an analysis part that determines the patient's hemodialysis status by measuring the blood urea nitrogen concentration before and after hemodialysis. It consists of a lab-on-a-chip that can sense the analyzed results and operate the entire system. The hemodialysis parts consists of an inlet part where arterial blood flows in before dialysis and an outlet part where venous blood flows out after dialysis. And there is a hemodialyzer that contains the first core technology of this artificial organ. The membrane used in this hemodialyzer has a uniform pore size of several nanometers and has excellent filtration performance. The dialysate supplement part consists of four main components: forward osmosis exchange filter, concentrate dialysate cartridge, spent dialysate inlet part, and concentrate spent dialysate discharge part. The principle is that spent dialysate meets concentrate dialysate in a forward osmosis exchange filter, supplies water to concentrate dialysate by osmotic pressure, and it works by being released into the renal tubules. And with this function, the size of the artificial kidney device can be dramatically reduced. Through Inicep's innovative Pioneer technology, patients will be able to get back to their daily lives, spend time with their families, travel, eat and drink freely, and return to a healthy life.
Yeah, thank you.
Nice.
Yeah, so a few comments from me, like this is a try to implement the sort of a miniaturized hemodialysis system that still utilizes the hemodialysis.
Now that will be inserted inside the body, the kidney,
Well,
or it is attachable outside.
If we watch their website, they make like the two words that basically suppose that this apparatus can be implantable or attachable to a human body. And that's basically, I think, a very good commercial move, because a few comments from me. Since I worked with the dialysis regeneration fluid a lot, it's still a big mystery for me if they implant this system in the human body and they have a concentrate of the dialysis system. And for instance, this internal apparatus said that the running out of the concentrate, what amount of this concentrate they have to prepare to actually maintain the continuous work of this apparatus during a prolonged period of time. We're talking about years. Would it work for a month or for a year, etc., and so on? So yeah, it's just a promotional video, and perhaps we basically do not know all the details of the project, because the primarily field of expertise is actually creation of the filtering membranes that they are going to use in the future apparatus. But the usage of the dialysis fluid is actually a headache on its own, and I'm just talking from the perspective of my work that I did with the different wearable artificial, this different dialysis regeneration units. Even if we're talking about the attachable or wearable artificial kidney, and if we introduce the dialysis fluid in this manner, and this dialysis fluid interacts with blood one way or another, so it could be a peritoneal dialysis where we just pour this dialysis fluid in the peritoneal, the abdominal, and after the interaction with blood we have the spent dialysis solution that we have to regenerate for the further usage. Or it would be a hemodialysis system where we have the extracorporeal mass exchange unit, and we still have the problem of regeneration of this dialysis fluid. So it's not exactly clear how they are going to implement the dialysis technology in terms of the implantable system, and how exactly they are going to constantly regenerate or renew the dialysis fluid in that particular case. So it's just a promotion video. They presented this particular technology, and they won the award for this innovation. So it's quite a promising project that basically has all chances to become like an available renal assistant device for patients who suffer from chronic kidney disease. But from my perspective, there are some white spots that are not completely clear in the consideration how the specific parts of this apparatus will work. That's just my opinion, so you can take it with a grain of salt. But basically, yeah, if we talk about the total implantable artificial kidney devices, we can consider this product, like the implantable attachable artificial kidney, as the potential candidate that will be a total implantable artificial kidney that basically does not exploit any biological tissues. And why exactly it would be the advantage is because we don't have to concern the survivability of these particular cells within the organism. We don't need to take into consideration the immune response to these biological tissues, because the immune response actually makes additional engineering and construction pressure in the sense that we need to create additional membrane that will separate this biological tissue from the patient's blood, and thus we will prevent the immune response. So basically, what's the great advantage of this particular approach is that they just use the membrane technology. Sorry. And the hemodialysis approach, without making this technology more expensive and more complicated when it's not needed. So yeah, on the current perspective, like there are multiple projects that related to the development of the implantable artificial kidney. Of course, due to different reasons, the attempt to imitate the rat system and the bar system, that means the usage of the biotechnology, is quite, yeah, that's just the scheme of what exactly they're going to do. It's the most popular technical decision that people are trying to implement. This goes for the kidney project, the kidney project, and other projects that implement the usage of hemofilter and biotechnology. But at the same time, there are some perspectives of using the total artificial kidneys that will not utilize any biotechnology whatsoever. So the perspective of what particular approach will become the first in the market is just a matter of time, and it's very hard to predict really what kind of technology will basically enter within. Yeah.
How much time duration this filter works means is any or fixed time? Is any fixed time? How much time duration this filter is usable in this artificial kidney?
Well, it's supposed that this filter will be used continuously for a very prolonged period of time, because there is no use in implantable technology that you need to extract next day, because you will hurt the patient more than you help him. So when we talk about the implantable artificial heart, or in that case about implantable artificial kidney, at very least we have to concern that this particular device have to work for at least a few years straight. Usually when we talk about implantable artificial kidney devices, for instance, if we take Sputnik that was developed by MEAT and particularly by Medical System Institute, this particular device was developed for the prolonged usage period for about 5 years or even more than 5 years. So in case of the implantable artificial kidneys, there should be considered the option of the prolonged usage of these membranes, and the membranes will be developed accordingly to perform the blood filtration for a very prolonged period of time. Well, at least 5 years.
Okay.
Yeah. So we still have some time, and I just wanted to make a quick overview of the literature that I used during the preparation of this course. It's not restricted just by this particular textbooks and that cover, well, because I used multiple papers, but if we're talking about some basic information that you can use as the foundation of the further research work, and if you're interested into diving into that, then I think that I would recommend the handbooks and of the dialysis and textbook of peritoneal dialysis. And what's good about those books is that handbook of dialysis is a book that with simple language explains the very foundation of the dialysis technology without going way too complicated to the different equations. So it basically gives a very quick, not quick, but very thorough and at the same time very understandable.
Comprehensive.
The principle of how hemodialysis works, the principle of what clearance is, how we estimate the efficiency of the dialysis, what particular modalities exist within the hemodialysis or peritoneal dialysis. So basically it's a very starting point to understand the principle that lies behind the hemodialysis or peritoneal dialysis. The textbook of peritoneal dialysis is more like a book for for doctors who perform the peritoneal dialysis. Nevertheless, this textbook provides a very good overlook on the mathematical equations and mathematical modelings of how the solute flux and how the water flux works during the peritoneal dialysis. So specifically chapter 4 and chapter 6 are very handy chapters for the engineers who want to know some basics behind the mathematics of this particular process. Of course, it has a lot of medical terminology, so if you want to dive into some specifics of the structure of peritoneal membrane, it's also a good book, but from engineer standpoint, I think that mathematical models that provided in this textbook are more essential for engineer than some physiological background. But just like we mentioned at the first lecture, in order to understand what particular process we are trying to module, we need at least some very basic outlook what biological system we are trying to imitate with mathematical module. And for that regard, I think that this textbook provides both the medical information and mathematical explanation of what exactly happens in that system. Anita Taylor and mathematical modeling in renal physiology is basically a very handy book for people who do math and engineering, because it provides some very basic outlook of what kidney is and what particular nephron structures are all about, what particular cell cultures they have within. And it has a very thorough and very detailed mathematical module of different fluxes that take place in there, the active ion transport, the working of the Henle loop, the working of the proximal convoluted tube as a whole, the primal blood filtration that takes place in the Bowman space, etc. So basically, it's very hard to cover all mathematical details within a very short course that's basically about the implantable artificial kidney devices. Nevertheless, if you want to use some mathematical background for the further development of that system, I think the good starting point of understanding of what exactly you need to imitate in the implantable artificial kidney is to understand of what exactly happens in the natural nephron. And the mathematical explanation is quite precise here, it's quite thorough. They have a lot of papers linked in that book, so it's a good starting point for that matter. Van Gelder, Steps Towards a Durable Artificial Kidney, is a very magnum opus work of this brilliant scientist who was working on different approaches of dialysis of regeneration fluid, and that's basically his very grand work on different complications and different approaches that take place during the development of the durable artificial kidneys. He also covers the idea behind the implantable artificial kidneys, and he explains thoroughly all complications that take place during the different stages of development. Like, for instance, if we talk about the dialysis regeneration unit development, he covers all approaches that have to be taken into consideration in terms of the construction of the durable artificial kidney. He explains all complications that take place during the different stages of dialysis regeneration, what particular problems occur during the electrochemical oxidation approach or enzymatic approach, So basically, if we talk about the different steps of the development of wearable artificial kidneys or some perspectives of implantable artificial kidney technology, it's a very good leading. And since digital twins and artificial intelligence was not like my primal field of expertise, I have to link another very thorough and very detailed book about the subject of digital twins technology that can be applied in very different subjects like weather forecasting, like industries, like the manufacturing of different machine details. It covers a lot of examples of how exactly digital twins are applied in biomedical systems, how they are applied in medicine, drug testing, how they are applied in engine testings and in industries, etc., and so on. So basically, if you want to dive into the digital twin and you need some starting point of what digital twin is and what is the connection and application of artificial intelligence technology in the digital twin development, I think a good starting book is the book provided by National Academies of Science, Engineering and Medicine, Foundational Research, Gaps and Future Direction for Digital Twins. A very good reading, and basically this is the main literature that I used during that course, and I think we have some time to address some questions from the audience.
Yeah. Maybe. So please ask if you want to ask any question, because this is the last lecture session is over. So maybe or you can ask then after the valedictory session also, because today is about to my valedictory session. Already it is I think 1:30. So maybe. Okay. So I will let me conclude the session.